Students, we are discussing the chapter Principles of Inheritance and Variation, and today we will discuss what is a trace cross. In our last class, we had discussed the law of segregation and back cross. So, trace cross. The cross between F1 hybrids and double recessive homozygous parents is called trace cross. So F1 hybrid means, if we consider the stem height character, the F1 hybrids are capital T and small t. The plant will be tall, but its genotype, phenotypically plant is tall, but its genotype is a hybrid. That means mixing of two genes, capital T and small t, two contrasting genes are present here. So when F1 hybrids are crossed with double recessive homozygous parents, or when F1 hybrids are crossed with the recessive parents are always homozygous. So it is called trace cross, small t. So this is trace cross. And the results, capital T, small t, gametes, small t. So I have shown this cross in back cross also, capital T, small t, capital T, small t, small t, small t, small t, small t. So the trace cross is performed to determine whether the individuals exhibiting dominant character are homozygous or heterozygous. So phenotypically this plant is tall, but we don't know whether this is homozygous tall or heterozygous tall. So to know whether this tall plant is homozygous or heterozygous, this cross is performed. Okay. So here the trace cross progeny, these two are heterozygous tall but heterozygous tall and homozygous dwarf. So its genotypic and phenotypic ratio 1:1. Both are same. 2 tall plants, 2 dwarf plants. Its phenotypic ratio. 2 heterozygous tall, 2 homozygous dwarf. Its genotypic ratio. So 1:1 ratio for genotype and for phenotype also. So here 50% individuals or 50% progenies are heterozygous dominant and 50% are homozygous recessive. So it gives 1:1 phenotypic ratio. Some non-Mendelian inheritance. So we have discussed two laws of inheritance, or Mendel's two laws, law of dominance and law of segregation. Now we'll discuss some non-Mendelian inheritance. So what is non-Mendelian inheritance? So there are several phenomena detected by the post-Mendelian geneticists which do not follow Mendelian principles of inheritance. So Mendelian laws of inheritance or Mendel's laws of inheritance, बहुत साइंटिस्टे এটাক ফলো কৰা নাছিলে আৰু মেণ্ডেলৰ পিছৰ বহুতবিলাক সাইন্টিস্টে কিছুমান ফেনোমেনন নেচাৰত তেওঁলোকে ডিটেক্ট কৰিছিলে which do not follow the Mendelian principles of inheritance. Mendelian inheritance সেইবোৰে নেচাৰত ফলো কৰা নাছিলে, and accordingly they put forwarded some laws which are known as non-Mendelian inheritance. So 3 non-Mendelian inheritance are discussed in your book, and these are: number 1, incomplete dominance; 2, codominance; 3rd is multiple alleles or multiple allelism. So these 3 non-Mendelian inheritance. So these 3 non-Mendelian inheritances are discussed in your book. So they do not follow the Mendelian principles of inheritance. Okay, so here we will discuss one by one. Coming to the first one, incomplete dominance. So what is incomplete dominance? In the post-Mendelian period, it was observed that the law of dominance was not universally true. Several cases have been recorded where a dominant character does not mask completely the expression of the recessive characters in F1 hybrids. Therefore, the F1 offsprings show a blending of paternal and maternal characters. This phenomenon is known as incomplete dominance or partial dominance or intermediate inheritance. So what happened? In the post-Mendelian period, the law of dominance was not universally true. So it was observed by a few scientists or geneticists. So they found several cases where they recorded that dominant character does not mask completely the expression of the recessive character. But according to the law of dominance, dominant characters are always expressed, and according to the law of segregation, we know that there is no mixing or blending of characters in nature. But the post-Mendelian scientists, they found that mixing of character occurs in nature. So this phenomenon of mixing of 2 characters, one is paternal character and another is maternal character. So this is known as incomplete dominance. নামটোৰ পৰাই গম পায় যে ইয়াতে 2টা ক্যাৰেক্টাৰৰ মিক্স হৈ হিহঁতৰ মাজৰ এটা ক্যাৰেক্টাৰ এক্সপ্ৰেছ হয়. That is incomplete dominance. So here dominance is not complete. Here the dominant character cannot completely cover the recessive character, and hence a blending or mixing of character occurs, and in the offspring a mixed character is expressed. So here I am giving one example. Incomplete dominance is well evidenced by the F1 hybrids produced in cross between homozygous dominant red-flowered snapdragon plant and homozygous recessive white-flowered snapdragon plant. So I am showing how. So in snapdragon plant, homozygous dominant red-flowered snapdragon plant. So it is, as this is the dominant character, red-flowered, capital R, and white flowered. This is recessive. recessive and dominant. So when these are crossed, these two, then the gametes will be capital R, smaller, smaller. Again, in the F1 hybrids, all the plants capital R, smaller. I am showing here roughly capital R smaller. Okay, so this is F1 hybrid. So here, according to Mendel, all the plants should be red in color because red is the dominant character, isn't it? So guteketa plant red hobo lagisil, but in nature it is found that all the plants in F1 generation all are pink colored. Okay, so this color is between red and white. When we mix red color with white color, a pink color is generated, isn't it? So here, instead of red, in F1 generation, pink colored flowers are produced. So it is proved that the red color is not completely dominant over white flower. So the heterozygous offsprings, so these are the heterozygous offsprings of F1 generation always produce pink colored flower. And the F1 hybrids then produce F2 progeny with identical phenotypic and genotypic ratio 1:2:1. So in F2 generation we'll get capital R. You know how to find out the F2 generation from F1 hybrids. So this is self-breeding of F1 hybrids, and the results of this cross I am showing here, capital R, capital R smaller, then smaller. So here red, pink and white. This will give you phenotypic ratio 1:2:1. Similarly, its genotypic ratio is also 1 dominant, 2 hybrid pink and 1 recessive character white. But according to Mendel, the phenotypic ratio should be 3:1 because all should be red and only one white. But in case of snapdragon plant, the phenotypic and genotypic ratio both are same, and it is 1:2:1. Clear? This is incomplete dominance.